speaker
Thank you.
is Professor Wenz.
Thank you.
Thank you very much.
Okay.
Hello, good morning.
Good morning.
Let's introduce your situation. Dr. Wenz achieved his PhD in food chemistry and technology in 1999 at the National Source University. I cannot read the original text you provided because it's in the language of Argentina. The university, the National Source University, is right?
Yeah.
Yeah. Since his postdoctoral stage at the Institute of Biochemistry, he's working now in biophysics of membranes, dealing with the architecture of the membranes, their physics behavior, lateral organization, and segregation of the lipids and the influence of these properties. In the last few years, he incorporated the near-infrared spectroscopy as an innovative tool to examine the water in membranes. The title of his talk is the Near-Infrared Spectroscopy and Multivariate Analysis for the Study of Water in Lipidic Membranes. Okay, now please, Professor Wenz.
Okay, thank you for the presentation. I think it's too much for me. And I want to thank you also to Professor Muncan and Tenkova for inviting me to offer this lecture, which is very simple and short compared with the Professor Di Salvo's talk. I want to show you an approach that I have started a few years ago, around 3 or 4 years, with the near-infrared spectroscopic to study the water in the neighboring, the surrounding water around membranes. This is a simplified depiction of a membrane where we have, as we all know, the acid chains, the hydrophobic part of the membrane, and the polar head, which represent the hydrophilic part, very well explained by Professor Di Salvo. And I was interested in studying what happened with the water in the interface between the bulk water here and the polar head of the phospholipid. For years, I was working with the fluorescence spectroscopy, and after a while, after reading some papers of Senkova, I say myself, why not to start to study membranes with near-infrared spectroscopic and aquaphotomic? Because of that, I jumped to this cliff and start to work on it. This is a simplified scheme of the approach I propose. We prepare artificial membranes. We make liposomes. I guess most of you know what it is, where you can handle the type and the concentration of the phospholipid to use to prepare the membranes. Once I have the membranes, the liposomes, I take the near spectra, a change in temperature, and then we apply some multivariate analysis. In this case, maybe one of the simplest one, that is principal component analysis on the matrix of the sample and the wavelength. And quickly, this is the result we found. This is the 60 spectra obtained from the liposome at change in temperature and at varying concentration of 2 lipids. And what we are trying to measure here is how the polar heads of the lipid affect the water that is close to the polar head, what Di Salvo said about the interfacial water. This is what I'm trying to disclose, to study, using, as we all know, the first overtone of the water, which is around 1,400 nanometers of wavelength. And this is based on the fact that the less the strength of the hydrogen bonds, the more the strength of the covalent oxygen and hydrogen bond. So what we expect when these hydrogen bonds debilitate is to observe a shift of the main band toward the left, so to shorter wavelength. That means that we need more energy to capture the vibration of this covalent bond. Conforming this is weakened, this became stronger. So this is what we expect. And this is the shift we employ to study the water and its hydrogen bonds around the polar head of the membrane, around or inside. Let's go straight to the results. This is the PCR output, where more than the 99% of the variation of the whole data was able to explain with only 2 PC, with only 2 principal components. Here, in PC1, for instance, we have the variation of the temperature from 13 to 58 degrees. So each of these samples, let's say the blue one, the blue line represents the water, the pure water, heated from 13 to 58 degrees. This variation in the spectra is captured mostly by PC1, which explains 97% of the variation. And the other lines, the green lines and the red lines, represent the effect of the phospholipid on this interfacial water. For this, we need another PC, which account around for 3% of the variation. So in all this, in this plot, we have almost the totality of the variation in the spectra due to the variation in the temperature and the variation in the composition. Here we can see how the effect of the phospholipid made some change in the spectra. The second, and maybe the second, another useful output of the PCA, of the principal component analysis, is the loading. And what do they tell us about? It tells about the wavelengths. Here, the farther a loading of a wavelength is from 0, so close to -1 or to 1. This is the, let's remember that loadings vary between -1 and 1. The farther loadings of a given wavelength is from 0, the greater the variation in the absorbance at that wavelength. Let's say, for instance, if we plot loadings against the wavelength of the whole spectra, this is the near spectra from 1100 to 2300, we see that at this wavelength is the place in the spectra where absorbance vary most. And also this one. These loadings represents the wavelength at which we can find the more variation in the spectra. And then, if we compare together the score plot and the loading plot, we can make some kind of zoom of the spectra, especially when they are very overlapped, and we want to disclose to see what happened with the effect we are measuring. We can make something like the typical biplot. But when you have too many variables, like here, we have around 1,200 variables, it's impossible to put in one wee plot both data. So another way is this one, is to compare in a separated plot scores and loadings and analyze the following. For example, if we take into account these samples on the left of the score, these are samples around at the highest temperature. And they have on PC1 negative scores. This one on the left. If we go to the loading plots and look for those wavelengths that have negative loadings, let's say this one, around 1,391, we can ascertain that these samples have or show the highest absorbance at this wavelength. In other words, this means that this is the wavelength at which the absorbance increases most and corresponds to these samples. So, on the contrary, these samples, the coldest, those samples that are on the right of the plot of the graph, they also have a positive score values around between 1 and 1.2. If we go to the loading plot, we see that the positive loading corresponds to this wavelength. 1546 is the absorbance at which vary most, and is this wavelength corresponds to the samples at which absorbance are higher, and this corresponds to the coldest temperature. So this means, what does it mean? It means that conforming temperature is going from the coldest values to the hottest value, absorbance is increasing at this wavelength and decreasing at this wavelength. This means that the main overtone of water is moving to the left, is moving to shorter wavelengths, around 1391, and the other one. So what does this tell to us? This tells that this variable temperature is moving our band to the left. And what happened now if we analyze the effect of the phospholipid? This is described in the score plot by PC2. And this move from negative value for the water, increasing values, conforming we are adding increasing amount of the lipid. If we want to compare this in the loading plot, we have to look to PC2, which is the one that describes the effect of the phospholipid. And this, we found here, the maximum around, this value is on the left again, on the left of the main overtone of the water. This also means that incorporating or adding lipid to the membrane, the water overtone moves to the left, as what happened when we increase temperature. This is the conclusion of the analysis of this simple output of the PCR applied over the spectra. A second approach we made on this spectra is to subtract, remove the signal of the bulk water on the spectra, removing the pure water from any of the lipid-containing samples. This is the raw spectra. After removing the corresponding water, pure water spectra, this is what we have, where we can clearly see that there are two populations of spectra. One of them each corresponding to one of the lipids. So, and what does it mean? This means that what we are reporting, what we are seeing here, is only the spectra coming from the molecules of water close or in the neighboring of the polar head of the phospholipid. This also we can see on the score plot. After that, we apply a new principal component analysis over the different spectra, and this is what we obtain: a score plot with a very clear separation of samples. One group of points is on the right and corresponds to this lipid, and the other one corresponds to this lipid. Oh, sorry. Are you seeing the screen?
No. Yeah, you should see it. Yes.
Something happened.
Okay.
We are on time? No.
Yes. Okay.
Yes.
Okay. Let's see this, and this is what we think about what is happening behind these experiments. in pure water, we have the water with 1, 2, 3 or non hydrogen bonds, and conforming we are incorporating lipids, some of those water cannot make so many hydrogen bonds, especially in this lipid, in DMPC, which has this polar head, which lacks of a hydrogen bond, an hydrogen, like it has the DMPA. This is the what we hypothesize about why this lipid moves the water overtone more to the left than this one, and more to the pure water, for sure. Okay, thank you very much for your attention. I guess we are on time.
Thank you. Yeah, we have time to have a discussion or comments. Anyone has problems?
Okay, can I start with the question? Thank you very much.
You're welcome.
Can you please show the PCA score plot? PCA score plot. Oh,
This one.
Okay, yes, thank you. Now, 1385 is related to protonated water clusters.
Yeah.
What is your comment about protonation in the whole story, if you have any?
No, this is a preliminary experiment, and I'm only seeing the whole picture. I have not enough data to say about several species of water.
Yeah, but my question was from a point of view of lipids, the relation between lipids and protonation. Maybe we can think about this. And also, from my side, I would say 1391 is a dehydration band, or we call it trapped water. So it means that this is the water that goes in between the lipids. It is beautifully seen here. So I was so happy to see this. But I don't know how we can, maybe Disalvo can help us with this protonation stuff, because it's obviously there is something happening to the protonation. I don't know, maybe Elena knows more about, do you remember about something about 1546?
Yeah. Yes, I actually also wanted to comment on this. So you have combination of these bands, 1546 and 1391. 1546, I think, is one of these strongly bound water types, which we saw yesterday from Professor Tonauer's talk, that there are various types of these strong bonds. So I think this is one of those types. And in this case, as temperature increases, there is a decrease at this wavelength. So this means that basically the water is being removed from this engagement in stronger bonds, and it is more involved in hydration of solids, charged solids like ions. So these ions then, this is what we see from 1391. I think we have this trapped water within, in between hydration shells of these ions and protons. Now, it is very interesting, for me, it was very interesting to see that you have also one more negative band of PC1 around 1800. So this is something that we connected very much with the water activity, with the actually mobility of water species. And this is a different type of information that we had from the first order. So it is beautiful that you see it here together. And I think that around 1800, this is where you can see proton mobility, or basically fluidity of your system, water activity in the most.
Yeah. Interesting observation.
I never saw, so it would be nice if I can know the band around 1800.
Okay. We discuss later. Okay.
Sorry, thank you. I know I took time. Thank you, beautiful lecture.
Okay, thank you, Professor Wenz. Now
Thank you very much.
to the third speaker of this session, Professor Zhang. Professor Zhang is a professor at the School of Pharmacy of Shandong University, China. He is currently the Deputy Dean of the Institute of Pharmaceutical Regulatory Science of Shandong University, the Director of the Key Laboratory of the Pharmaceutical Preparation Technology Research and Evaluation of China National Medical Products Administration, the Chief Scientist of the China's National Key Research and Development Program in 2019, and won the third 陆婉珍